So in this video, we'll be looking at characteristics of eukaryotic cells, so human cells characteristics, and which aren't microbes, of course, but also the protist groups would fall into this category, along with the fungi. So fungi, protist groups, they are all eukaryotic cells. Let's take a look at them. And I should warn you, strap in. This is a fast ride through them because these are cells that you've learned about in other classes, and so I'm going to skim across the top a little bit more on this one. So, kingdoms Animalia, Fungi, and Plantae are all the protist kingdoms, and microbiologists, they study the microscopic algae and protozoans, the fungi, those are the protist groups, by the way, the fungi, and then among the animals, the microscopic and macroscopic parasitic worms are also of interest. The cells of eukaryotes are larger and much more complex than prokaryotic cells are, and they do have membranous organelles and a nucleus with a membrane around it. So the DNA is protected inside a nuclear envelope. And rather than having circular DNA, all the DNA that we find, the eukaryotic DNA that we find in eukaryotic cells, are straight chains of DNA rather than being circular pieces of DNA. So, as we said, the internal structure is much, much more complex than in prokaryotes. It has nucleus, and then there are other organelles, the mitochondria, chloroplasts, if it's a photosynthetic organism, ribosomes for making protein, the endoplasmic reticulum. I kind of think of this as the workbench that the ribosomes and other enzyme workers are building molecules on. The Golgi Apparatus, which, if we're thinking about a factory or a workshop as our metaphor for the cell, then the Golgi Apparatus is where we finish the products that are being made. And so here we find enzymes that are busy cutting up and reassembling the proteins, the carbohydrates, and the lipids that were built at the endoplasmic reticulum, and then packaging them up into a vesicle, so wrapping them up for shipping either throughout the cell or outside of the cell itself, outside the cell. And then some of the vesicles that we find inside of a cell include lysosomes, peroxisomes, and then vacuoles, which are long-term storage. And then finally, we also see lots of cytoskeletal elements, so microfibers and microtubules. So let's start with the nucleus. The nucleus is surrounded by a double layer of plasma membrane called the nuclear envelope. And so here you can see one of the layers, and here's the other layer. So this nucleus, of course, would be very large. It would be, this big based on this picture. But you can see the two layers of nuclear envelope, and then every once in a while we'll see a nuclear pore that allows large molecules to enter or leave the nucleus, as well as allowing ions and other things that are wanted inside of the nucleus in. The nucleoplasm is the gel-like material that fills the nucleus, and inside of the nucleus we see dark staining spheres that are called nucleoli. So nucleoli, while it officially means little nucleus, that is not what it is. It's built primarily of RNA, one of the nucleic acids, and it's used for assembling ribosomes. So it builds the ribosomes, or at least builds parts of the subunits of ribosomes. And then, kind of spooled out inside of the nucleus is the DNA, the strands of genetic material. And just like in the prokaryotes, it is the code for the proteins that the cell needs to build. And for the most of its life cycle, the DNA occurs as chromatin, kind of unspooled threads that are just kind of laid out higgledy-piggledy inside of the nucleus. But during cell division, the DNA condenses, it spools up into the chromosomes that are we can see them under a light microscope. So you can see the chromosomes under light microscopes, but when the DNA is unspooled and occurs as chromatin, you can't see it. It just makes the whole nucleus appear dark. And it spools around specialized proteins called histones that are there to help, make it really, really contracted. And it's important to do that spooling up so that we can appropriately separate all of the DNA into the two daughter cells that are made during cell division. And there are two copies of each chromosome. So we talk about eukaryotic cells as being diploid because there's two copies, one that you get from one parent, your mother, and one that you get from the other parent, your father. So looking at reproduction in eukaryotic cells, there are two processes that the cells use to reproduce. There's mitosis, which produces two identical daughter cells that have exactly the same DNA inside of each one. So mitosis is the division of the nuclear material. And then cytokinesis is the activity that follows immediately after mitosis, where the rest of the cell divides. So mitosis divides the nucleus into two identical nuclei, while cytokinesis divides up the cytoplasm, and one nucleus goes to one cell and the other nucleus goes to the other cell. So we produce identical copies. They are diploid. Sorry about that. It produces identical diploid copies through mitosis. And this is how the body of a eukaryotic organism grows in size. We make two copies every time we do mitosis. Meiosis, on the other hand, is the process that is used to make the reproductive cells, to make egg and sperm. And in that process, we actually divide away from each other the two copies of the chromosome that we have inside each cell. And so through 2 rounds of cell divisions, we produce 4 cells. And in the processes of meiosis, we are able to separate the pairs of chromosomes from each other so that the cells that we produce are haploid rather than diploid. So they only have one copy of each chromosome in them. So we produce 4 haploid chromosomes, haploid gametes at the end of meiosis. And those gametes then later on fuse together to produce the new diploid offspring. So that's why we have to do meiosis. To take an egg and a sperm that had a diploid number, you would now have a tetraploid offspring. And so we have to reduce the number of chromosomes in the cells during meiosis in order to restore the diploid condition when the egg and the sperm fuse together. So through meiosis, we can produce either gametes, which then later on fuse to produce a diploid offspring, or we can produce a structure called a spore. And so this is a process that we see primarily in the fungi. And a spore is a resistant resting stage that can outlast adverse conditions, and later on it will germinate. So the spore then germinates and grows to produce a individual that then produces the gametes. So spores, they are a resting stage to outlast bad conditions, and when they germinate, they grow into usually a much smaller individual whose primary function is to produce gametes. So that's the nucleus. As we look around, we can find the energy-producing organelles in the cell. Mitochondria are one of them. The function of mitochondria is to convert chemical energy in the glucose molecule into chemical energy that the cell can use. And that chemical energy form is referred to as ATP, adenosine triphosphate. I won't test you on that. But we do this process of chemical reactions called cellular respiration, and in return, we get a bunch of ATP molecules. Mitochondria, the ATP producers, are a double membrane organelle. They have, so just like we saw with the nucleus, there is this double membrane. The inner membrane is folded up very, very tightly into folds called cristae. So the cristae are actually the folds themselves. So there's one cristae right there. Here's another cristae right there. Crista, I suppose, would be the singular. That gives surface area. That's all about providing additional surface area for the respiration, cell respiration enzymes that make all those chemical reactions happen. They also have their own DNA, interestingly, and it is circular DNA that reproduces not when the cell does mitosis or meiosis, but on its own little cycle. And that is one piece of evidence that suggests that mitochondria evolved from free-living prokaryotes that got brought into another prokaryote cell, and rather than being digested, was actually held there because of its abilities to make ATP. Okay, so those are the mitochondria, one energy-producing organelle. Chloroplasts are the other energy-producing organelle. Their job, rather than to make energy out from glucose, they take light energy and they use that light energy to make the chemical energy in the glucose molecule. So because of photosynthesis, and if we break down the name here, photo means light, synthesis means building, so we use light to build glucose. So they have the very cool function of converting the physical energy of light into biological energy in the chemical glucose. Also a double membraned organelle. The inner membrane is organized into these little pouches called thylakoids. So thylakoids, and they are stacked. These sacs are stacked up into these structures called grana. And the job of the membrane of the thylakoid is to provide surface area for all the enzymes that do the processes of photosynthesis. So once again, highly folded membrane, and it's all about surface area for the photosynthesis reactions to happen on. They also have their own internal DNA that looks very similar to prokaryotic DNA. It's circular too, and it will reproduce on its own cycle. Oh, you're hearing an alarm in the background. I forgot to turn it off on my phone. So it's going to play that lovely music. Sorry about that. So the chloroplast is also understood to have evolved from free-living prokaryotes that got incorporated into a host cell. We'll talk about that theory in a little bit. So next on the list are ribosomes. Ribosomes are organelles whose job is to make protein, and they do it using the instructions that are found on the DNA in the nucleus. But the DNA never gets to leave the nucleus, so the way that we do that is we make an RNA copy of the DNA. The RNA is allowed to slip out of the nucleus, and it finds ribosomes and then tells the ribosomes what to do. So they're found either free in the cytoplasm, and again they form those little strings of ribosomes like we saw in prokaryotes in the cytoplasm of eukaryotic cells, or they're attached to the membrane of the rough endoplasmic reticulum. So here we can see all those little green specks on that membrane that's right up close to the nucleus. That is the endoplasmic reticulum, and those green specks, or the little white specks, are the ribosomes on it. Endoplasmic reticulum, if we think about ribosomes as being a worker in our factory. Oh, and by the way, the chloroplast and the mitochondria, they would be like the power plant for that factory. So if the ribosomes are workers, along with the enzymes being workers, then the endoplasmic reticulum would be like the workbench where the workers stand and build their products. So it's this long and very convoluted network of tubes and sacs that are all built of plasma membrane. And their job is to make large molecules like proteins and carbohydrates and lipids, and then move these molecules throughout the cell. If it's a rough ER, it's rough because it has all these ribosomes attached to it. And you look at it under a microscope and it looks really, well, rough. And so with the the ribosomes attached, then its job is to synthesize proteins. Smooth ER doesn't have ribosomes, and so its job, because there are lots of enzymes associated with smooth ER, its job is to synthesize lipids and to build carbohydrates as well. So all of that stuff gets packaged up into a little membranous bubble called a vesicle, and we send it off to the Golgi apparatus. So the Golgi apparatus appears, like if you look at it in cross-section, it's like this stack of sacs built out of plasma membrane. And their job is to accept a vesicle coming from the endoplasmic reticulum and then to modify the stuff that's inside that vesicle and package it up into new vesicles that are then stored in the cell, or they get released from the cell so that the contents are released by exocytosis. That's a process we'll talk about in the next lecture. And if that's the case, then they're referred to as secretory vesicles because they secrete the product made by the cell from the cell. And last of all, they produce membranous stuff. So they produce new membrane, and then that new membrane can be added to the cell membrane or to other membranous parts of the cell. Lysosomes are one of the vesicles that are produced in the Golgi apparatus, and they're filled with enzymes that are used to digest nutrients and destroy foreign materials that enter the cell. So that prefix lys means to break up or cut up. Soma means body. So these are the digesting bodies, or we could think of them perhaps as the composter in a factory that's shredding everything and then composting it so that the nutrients can be used by another process in our factory. Peroxisomes are also vesicles that are produced by the Golgi apparatus. Ooh, I should have had apparatus written there. I'm sorry about that. So there are vesicles that are also produced by the Golgi apparatus, and they contain enzymes whose job is to digest nutrients and any dangerous peroxides like hydrogen peroxide that can build up in the cell. And there goes my alarm again. Dadgummit. And then last of all, vacuoles are usually larger vesicle kind of structures. They're membrane enclosed components that store up things that are like in deep storage. So we might store starch in a vacuole, or glycogen or fat, like a lipid droplet would be stored up in a vacuole. And in the plants and in some other organisms, it also is used to store water to help kind of maintain the cell shape. So it becomes this sac in the middle that you can fill up with water, and that kind of plumps up the whole cell. So those are vacuoles. Last of all, for the organelles, is the cytoskeleton, and there's two kinds of materials that we find in cytoskeleton. They're microtubules and microfilaments. Microtubules are just like what they sound like. They're like soda straws. They're composed of a protein called tubulin. They're hollow in the middle, and they're very structural, strong. They give rigidity to the cell, and they allow some cell movements, like during cell reproduction, as we're making the cell, or pulling apart the chromosomes in the cell. Then microtubules are involved with that. Microfilaments, these, rather than being hollow rods or tubes, they're solid. Microfilaments are built with the protein actin. So actin is the big protein that we see making up microfilaments. There are other cytoskeletal elements as well, but we'll just stick with microtubules and microfilaments. And again, they give rigidity and structure to the cell. They provide attachment sites for cell structures, and they allow movement both within the cell and of the cell itself. So lots and lots of organelles associated with